Hey everyone, my name is Janosch Linkersdörfer and I'm the co-director of the data center for the ABCD study. Calen Smith and I will introduce you to the ABCD release 6.0 and then give you an introduction to the data exploration and analysis platform DEAP. The 6.0 data release is the first release that gets released through the NVDC Data Hub. This platform releases data from both the ABCD study and the HBCD study and provides several tools to get access to the data and information about the data. The 6.0 data release was just released in June 2025 and then contains data through February 2024. That means that it includes about 50% of the 6-year annual visits and almost all of the previous annual visits. In total, the release includes 7 annual visits. 4 of those are imaging visits and 5 mid-year time points for the core study. And then in addition to that, it includes data from 5 different sub-studies. In total, the tabulated data set contains 83,000 variables. The 6.0 data release includes several important changes. For example, we improved drastically on the quality of the data dictionary and the included meta information. We now provide consistent data types for all of the variables, units where applicable, and more information about the assessment situation. We also include more administrative variables. For example, now we include timestamps and the age of the youth for every assessment. Another important point is that we now use a standardized coding scheme for categorical variables where all the standard responses like yes and no or don't know, decline to answer, use the same code across the whole tabulated data set. We introduced a new standardized ReproNim scheme, which I will explain in the next slide. And we now created all summary scores that are included in the tabulated data resource with a new R package that we wrote specifically for that purpose. And we extended our documentation website, which you can find under the link mentioned there. Finally, and importantly, we also changed the structure and organization of the file-based data. Where applicable, we try to use the bits standard to organize the files. We developed a new standardized naming scheme that we applied across the whole tabulated dataset. So all the 83,000 columns are renamed with this variable naming scheme. This naming scheme has 4 main components in which we encode the research domain, the source who provided the data, a table component and an item component. So for example, here you see one example from the mental health domain where the parent provided the answers to the short social responsiveness measure. And in this case, it's the item 6 in the measure. The table component and the item component can further have more keywords. So for the table component, for example, we encode subscales or sub-elements that are similar and give them the same keyword. Like in this example, the weight and the father components. For the item component, we also have a standardized scheme to denote don't know responses or longitudinal items. And all those details can be assessed in our data documentation. Speaking of our data documentation, this is the landing page of it. And this website provides a lot of important information. And I would really encourage everybody to read about the measures that we are about to use in the data resource. We have very detailed documentation for each of the included tables in the tabulated data resource. So at the top here, you can see an overview table for a given domain, and that brings you different links and gives you some basic overview of how many variables are included, at which events this measure has data. And then further down on the page, each table has a specific description, shows you the key references. And specifically, we now include a link to the data dictionary viewer that is implemented in our DEAP application that we will present to you in a bit. And where applicable, also a link to our documentation for the R package computing scores for a given table. Now we would like to introduce you to the Data Exploration and Analysis Platform, or short DEAP. The goals for DEAP are to lower barriers to doing science with NBDC data sets. We provide user-friendly tools to create personalized data sets and eventually explore the data resource using visualization and test any kind of hypothesis using state-of-the-art analysis methods. We do so by enforcing scientific best practices and providing guidance. And our specific goal for this application is also to foster collaboration and sharing and reproducible science. For that purpose, every object that can be created in DEAP, so currently datasets and later explorations and analyses, can be stored in their own section. Objects can further be organized by using labels or assigning objects to projects, which can be seen in My Projects. And then every object can be shared with collaborators. That makes the object available to the other person, but only in a read-only fashion. And lastly, objects can be logged. That is used to make objects not changeable anymore, even for yourself, and is useful, for example, when sharing an object as part of a reproducible paper. DEAP has different modules or functionalities. The first one is the data dictionary and ontology view. This can be used to explore the ABCD or HBCD data dictionaries and ontologies, and can be used, for example, to search the data dictionary for variables of interest, to view variable statistics, and so forth. This feature is available already and is available also to the public, so you do not need to log in to DEAP to see and explore the data dictionary. The second feature is the creation of individual data sets. This feature can be used to create any custom data set and download them in different file formats. This feature is also already available and is only available to users with a valid DAC. As for the planned features of DEAP that will be added later this year or beginning of next year, the first one is a data exploration module in which you can create your own customized plots and store them in the application, and then also share them or log them and share them as part of a research paper. This will give you a lot of flexibility to create plots exactly how you like them, change colors, fonts, and so forth. We will also add a data analysis module to DEAP. In that module, you will be able to specify inferential statistical analyses and run them directly in the platform. And this will allow you to run both standard models on the tabulated data, but also brain imaging models with those same models in a fast and efficient manner. We will make sure that the interface is designed in a way that makes it really easy for users of all skill levels to specify these models and run them in a correct manner that takes into account the complex nesting structure of the ABCD study. Lastly, DEAP will also allow users to work with the data in code-based environments. For example, they will be able to launch an RStudio session or a Jupyter Notebook to work with the data in R or Python.
Hi, my name is Calen Smith. I'm going to be going over the DEAP Exploration and Analysis Platform demonstration. Before I begin, I just want to note that you can access DEAP through nbdc.deepscience.com. Or if you're looking for a specific study, you can do abcd.deepscience.com or hbcd.deepscience.com. DEAP is compatible with both ABCD and HBCD datasets, and you can toggle between the datasets once within the platform too. So you don't need to use the specialized URL to access it. Accessing any of these URLs will allow you to go back and forth between the datasets. You can also access it via the QR code on the top right corner of the screen. Before I begin, I'd like to note that we have a robust documentation website as well. You can access that at the URL or the QR code on the screen too. We have an hour-long tutorial that's available on that website as well, so if you would like a bit more in-depth explanation, feel free to just skip this video and go to our website and access that tutorial there. Moving on to our demonstration now. Once you go to nbdc.deepscience.com, you'll be redirected to a website that looks like this. This is going to be the landing page for DEAP. On the left-hand side, you can see that I'm not currently logged in. The first part of our demonstration is going to focus solely on features that are available to the public, meaning that you don't need a data use certification to access any of these features. And then the second part of the presentation is going to focus on features that are only available to those with a valid DUC. Here you can see the feature that I was talking about before, where you can toggle between the ABCD and the HBCD study. Clicking over to HBCD, you can see the color changes so you know which study you're in. Our URL also changed to HBCD. From here, you can explore the HBCD dataset using all of the same features that are available on the ABCD side. I'm going to use the ABCD dataset today for the purposes of our demonstration, so just toggling back over there. Later on in our presentation, we'll talk about my datasets and projects. Those are going to be relevant only to users with DUCs. For now, we're just going to be using our exploration features, which are open to the public. So we'll start by clicking Create a Dataset. We're now presented with two sides of the screen. On the left-hand side, we have our ontology tree, and on the right-hand side, we have our data dictionary. These allow you to both explore and understand the dataset that you're utilizing better. We're going to start off with the ontology tree, but I'd just like to note that you can see here, currently we have 83,000 variables displayed in our data dictionary, but as we click down the ontology tree, that number narrows, so now we have 63 variables. The data dictionary is filtered and reactable to the ontology tree. So the ontology tree can be used as a way to narrow down the data that you're looking at to make finding the data that you want to select for your data set easier. I'm going to go ahead and get rid of our data dictionary for now. And then you can also notice that as I click back down, it's recollapsing the levels of the tree to bring me back to where I'm at before. You can also use the reset button up here to do the same thing. The ontology tree shows you the levels that which the study is organized in. So depending on which domains you're operating under, it will generally be the same, with the exception of imaging having two extra levels, which are metrics and atlases. So it always is going to start with the study type. So in the case of ABCD core and sub-studies, and then it's going to go domain, subdomain, the source. In the case of linked external data, it will always be like linked external data. Let me pull up a different one. And then here's a good example. Youth and parent, and then finally we'll get down to the assessment or the measure. All of these levels are reorganizable too. So if we're not using the imaging domain, we can go ahead and get rid of metric and atlas. And if we're most interested in parent measures, for example, we can go ahead and drag source up top. And then it's going to rearrange that ontology tree to the order that we are most interested in utilizing it in. Again, going ahead and resetting that tree. You can also go ahead and share the tree using this link right here. That's going to generate a URL that you can send to collaborators so they can see the same measures and assessments that you're looking at and copy out that same position. And then last, because our studies are so robust, you can kind of end up with complicated tree structures sometimes. So let's go ahead and click out some of these. Okay, so you can see that they get pretty big. If you ever want to just narrow down the focus to only the things that you're interested in looking at, you can go ahead and click this focus button. It's going to drag everything in to only show you the things that you've already selected, kind of narrowing out the noise. I'm going to go ahead and exit focus mode and then reset our tree so we are back to 0. It's showing all of our data, and then we're going to switch over here to the data dictionary table. On our data dictionary, you're going to have all of the columns that exist within the study data dictionary. So by default, it's going to show you the variable name, variable label, variable type, data type, level, measurement, and unit. The domain is something I had selected earlier, so that's not a default one. And then on the right-hand side, you'll see what the details column is. That's going to be able to show you all of the details about the variable that you're looking at. And we'll go into that in a little bit more depth in a second. The data dictionaries table is also highly customizable, so you can ascend and descend the columns by sorting. You can pin them to left if they're of interest and you want to have that always visible, no matter how many columns you have. Similarly, you can pin to the right. And then from this tab up here, you can show and hide columns, or as well as within the individual column settings too. You can see here, these are all of the columns that exist within the ABC Study Data Dictionary, so quite robust. You can also download all of these as a CSV, which I'll show you in a second, but you can display any of these that are of interest. So if you want to know what domain things fall in, you can go ahead and click that, and it will show you the domain that the variables that you are looking at fall in. We also have all of the old names in the data dictionary as well. So if you're familiar with the variable names from NDA or from DEAP 1.0 or even from REDCap, you can go ahead and display those as well. I'm going to go ahead and reset this again. And now we're going to talk about the search function. The search function works across all columns of the data dictionary, regardless of whether they're visible in your table or not. So if you're not quite sure where a variable is housed in the structure of ABCD, what its variable name is, etc., you can use this as well. So if we were looking for a variable that describes whether the participant is right-hand or left-handed, we can go ahead and type that in there. And then it's going to bring up any variable that contains the word handedness. So you can see that the variables related to whether participants are right-handed or left-handed are centered in the neurocog domain, use source, and then it's the Edinburgh handedness scale. Similarly, you can also filter or search through the data dictionary using our filter function. The filter function works on any column that's currently displayed within the data dictionary. So if you'd like to filter it, then you just need to come over here and add it to the view of the data dictionary. I'm going to go ahead and add in domain for the source of our demonstration, and let's go ahead and filter. So let's do a variable names. We want it to be in our MR domain, and then I'm going to do a structural MRI measure and look at a area. So now you can see that we're limited down to only variables that follow that similar structure. So they contain MR underscore Y underscore SMRI double underscore area. We have 288 variables shown right now. And I'm going to go ahead and show you the details view for one of these. The details views for all of our variables are going to be different depending on what type of variable you're looking at because they have different descriptive information about them and descriptive information in the data dictionary. Something unique about our MRI brain atlases is that you'll have this 3D brain viewer where you can click into it and look at the brain region that you're interested in. I'll show you a few different examples of this as well, but every detailed view is going to have the variable name, the variable label, some information about the variable, so the data type, variable type, any units, etc. You'll notice that the table name is hyperlinked. So if you click onto that, that will bring you over to the study documentation website where you can then navigate down to the actual table and learn more about those specific variables. We'll also have aggregate statistics for any of our variables. So it's summary table and then a summary chart, the placement of that variable within the ontology tree, and then any old names that existed for that variable are available in the name mapping section. Again, anything that you can see in the variables view besides the aggregate statistics is going to be available in the data dictionary itself as well by just expanding out these columns too. Let's go ahead and find a different variable now. Let's look for something that would have a good bar chart to show you our categorical descriptive. So let's go with our domain, and then we're going to do, our domain is in the general. And then something like education will be a good example for this because we'd expect it to have a semi-even distribution. Here we go. So in the aggregate statistics, we'll always have a table and a chart. For our categorical variables, it's going to be a summary of counts for the table, and then it's going to be a bar chart. For our numeric variables, it's going to be a descriptive statistic table containing information like quartiles, skew, things like that, and then a box plot for the chart. All of the time points that this variable has data at are going to show as the columns. So you can see this variable has data for our baseline through our 6-year annual time point. The complete is going to be out of the participants that have data within that table. So for participants that have data in this table, there are only 14 participants missing data for this variable at the time point. Let's go find a good example now of a numeric variable. I'm going to show you how the search function, the filter function, and the ontology tree can work in conjunction now. So let's look for a neurocog variable, for example. Let's limit it down to only show neurocog, and we only want to show task data because we're going to look for something that has reaction time in it. But let's look at the units now, and we'll filter by unit. And let's look for a variable unit that contains seconds. So now that's bringing us down to only things that contain seconds. As you can see, they're usually in milliseconds here. And if I'm looking for a specific task, I might now use the search function. So let's go ahead and look for the delayed discounting. So now we have all the reaction times that exist for the delayed discounting task. And you can see that you can use a function of both the ontology tree, the filter, and the search function. Find anything that might be of interest to you. This is going to be one of our continuous numeric variables. So you can see this one has data at the 1 year, the 3 year, and the 5 year annual time point. Different kind of chart display. This one is also mildly editable, so if you're not interested in quartiles, you can go ahead and remove those. And then the summary chart is going to be a box plot. That one might not be a great example. Better example of a box plot. As you can see, it mimics the information that's available in the table. One more example of variables that exist, or some things that exist in the data dictionary details view, is images. So if there are ever any instructions, headers, information that the participant would see, that's all available in the data dictionary as well. You can find that under instruction, headers, notes, and then in our pain survey, for example, there is an image where our participants can see a map of the human body and then select the region of the body where that pain occurs. So you can see any images that are relevant to them too. A few other examples of things that might exist within the details view or within our data dictionary include links out to the documentation, which you've already seen, links to our summary scores, ABCD scores website, as well as links to all of our data warnings. So if we ever have a warning for responsible use or for data quality, that will be listed as well. Those are also searchable columns in the data dictionary. So let me go ahead and reset. And then if we scroll down here, you can see data warnings by variable or by table. And then documentation for the summary score as well are all searchable URLs. So let's go ahead and filter to show the URL for the summary scores. You can copy paste this URL, or if you click into the details view, the variable type summary score is going to be hyperlinked, and that will take you to the information about that summary score on ABCD scores. Similarly, let's go ahead and look at a data warning really quick. Again, we can grab the hyperlink from here, or we can go ahead and click in here, and then there will be a hyperlink out to that specific warning on the documentation website for you to learn more about what that warning is. That is our features that are available publicly without a data use certification. If you want to be able to actually go ahead and click variables, compile them into a custom data set, download any data, you're going to need a data use certification. I'm going to move on though now to our features that are only available to folks with a data use certification, which is going to start by logging in. When you click login, you're going to get redirected to here, and then you'll just click RAS. And you're going to log in the same way that you did when you applied for the data use certification. Now we are logged into DEAP. You can see over on the left-hand sidebar, I'm now logged in. We're now going to go over the features that you can do with a data use certification beyond exploring the data sets for the study. So let's go back to create a data set. From here, we're just going to focus on creating a custom data set, and then we'll go over pre-assembled data sets in a second. Custom data sets have a limit of 10,000 variables. The pre-assembled data sets contain anything up to all of the 83,000 variables that exist within the tabulated resource. So if you're needing more than any of the 10,000 variables, I would suggest looking at our information about pre-assembled data sets. All of the features that I've been over previously work in the exact same way. So filtering down, clicking into the details view, modifying the data dictionary, things like that. Let's go ahead and create our data set though. To create our data set, let's first identify a survey that we want to grab the variables for. Let's say we want to go find the variables for all of our blood biospecimens. I'm going to go ahead and type blood into our search bar. You can see that we have some variables from our linked external data that have the word blood in them. That's not quite what I'm looking for. So I'm going to go ahead and search by our variable name and say that our variable name has to start with pH because I know that our blood variables fall within the physical health domain. That then limits us down to 87 variables, which seemed like about the right amount. So from here, we can either go ahead and select variables individually and add those to our data set, or we can click here and select all of the variables that are displayed on the current page. You have to be careful with that though, because currently I have it filtered to show 100 variables per page, but if I had it on 25 and I select that, it's not going to go ahead and select all the variables that I have it filtered to, just the ones that are on the front page. However, if you would like to select all of the variables, regardless of whether they're visible on the page or not, you can go ahead and click select all. Again, though, that's only going to do the ones that you're filtered down to. So whether you have your data set or your data dictionary constrained by the filters function, by the search function, by the ontology tree, it's only going to grab the number that is visible down here at the bottom. So let's add those to our data dictionary or our data set. And then let's go find one other one. Maybe I have an old data set where I've used our variables in the past. If you select this little clipboard right here, it'll give you the option to paste a list of names in there using the old naming functions. For HBCD, they only have one release, so only one naming scheme. So for ABCD or HBCD, if you just selected this variable name, you can use the current variable names and paste a list in there. We're going to say that I had one from the old NDA naming scheme, though. I'm going to go ahead and paste that list in there, just get rid of these bullet points really quick, and then go ahead and hit submit. You're going to see that we got an error. It will point out anytime that any of these are invalid. It won't go through until only valid entries are remaining. You can either select up here to show only errors, or if you scroll through the list, all the errors will be highlighted in red. Variables might not be there because they might not be the correct names. There might be a typo. It might be the wrong naming convention. Perhaps I accidentally meant to put REDCap instead of NDA, or it's a mix of variable names. Or it might be that that variable does not exist in the data release for some reason. If you're ever unsure, just go ahead and feel free to submit a ticket and we can help check to see what the error might be. In this case, it's a typo, so I'm just going to go ahead and get rid of that and then click submit. And now our 7 variables have been selected for us to go add to our data dictionary. Let's go ahead and click add. And from here, we're going to go to edit and save. We can name our data dictionary. So let's go test, demo, ReproNim. And then we can go ahead and edit our variables. So if we want to get rid of any variables, for example, let's say we decide that this variable and this variable are no longer relevant. We can go ahead and click remove for those. Over on this side too, you can modify the display the same way. So look at any of the columns you might be interested in, look at the details view, etc., and filter down the same way. When you filter or search down and then select variables to remove, it acts the same way. So it's only going to select the ones that you have it filtered down to remove, as opposed to just selecting all of them. Once we're happy with our variables, we have 92 variables. We're going to say that's okay. We're going to go ahead and hit Save Dataset. And from here, we're going to get an additional variables pop-up. These are all variables that have been identified by the study experts as being useful for any data set, regardless of what your question is. They'll have some pre-selected out for you, so you can see that they've selected some key ones that they think any data set would find useful. For example, the age at the start of the visit, the family ID, the assessment site, things like that. If you are wanting to use any of these variables, you can go ahead and click and select any of them. You can look at the details view to learn more about them, toggle through them. In our case, though, we're just going to go ahead. Actually, we will add a few of them. Let's add all of the ones from the design and nesting and click save. Now that our data set is done, we're going to go ahead and view our data sets. Another way to get here is to go to the My Datasets tab. You'll see all of the datasets that I have created. Here's the one that I just created. We have some information about all of the datasets that we have. So variables, if it's sorted into any project, the release it's from, if it's been shared, creation, created, etc. You can modify the columns that are created on this or shown on this table up here. You can filter down your data set similarly to how you filter variables, and then we'll talk about the archive function in a second. When wanting to learn more about the data sets that you have or edit them, you can click this little eye icon. It's going to pull up all the variables that you have selected in the data set in which you can toggle through them, look at the variable details, change the name, edit the labels and projects, etc. And then if you want to edit, you can just click Edit Dataset. It'll bring you back here. If you make any additions, you can go ahead and add those and then update your data set. Now let's talk about downloading our data. So if you click this button right here, it's going to give you the option to download. We have 7 formats that you can download the data in. And depending on the format that you're downloading in, you might be able to make some customizations, such as displaying categorical variables as values or labels, and then coding the missingness as NA or null. We're just going to leave it as the default for now and go ahead and select CSV and click Prepare Dataset. Now over here on the left-hand side, you can see it's working on preparing that data set for your download. It will prompt you when ready. While we wait for that, let's go ahead and go over some of the other features that exist in my data sets. So you can see here, some of our data sets are tagged. Those are going to be what we call labels. You can add labels either when you're creating a data set or if you come over here and go Edit Label. You can click any of these to add them as well. Let's go ahead and add another label too, just so you can see how that works. Now that we save that and come back here, you can see that all of our labels have been assigned to our test data set. These are also assigned into projects. Projects are essentially folders where you can store data sets that have an alike use or alike content. Same place, so you'll come here, click Edit, Projects, and from here you can select it into any of the projects, or you can come to the My Projects tab, click into any of your projects that exist and add it there as well. So you can see we haven't added it to this bottom one yet. So let's go Edit Datasets. and add our new data set in there, and it's now populated over here. Going back to the My Datasets function, you can also see the share function. You can see that these bottom ones are a dark gray and the top is a light gray. If you need to send a data set to somebody that you're collaborating with, if you select this button right here and click Yes, Share Dataset, it's going to generate a URL for you that you can send to them, and it will open on their DEAP platform once they've logged in. You can see that it's turned to a dark gray. So a dark gray indicates that it's been shared, light gray indicates that it hasn't been shared. And then if that arrow is pointing down, or if it has a different name in the created by column, that means it was shared with you. Your collaborators are not able to edit your own data sets, but if you ever need to use a data set that's been shared with you, or they need to use yours, you can always duplicate the data set and make your own modifications to it. Although they will see any appending changes that you make to the data set, any updates, things like that. So you can stay in sync with any data sets that have been shared with you, but just not make changes in sync with each other. Looks like our data is ready now. So when we go ahead and click download, it's going to re-prompt us over here and we can select completed. That's going to download our data set, and I'm going to go ahead and open that. And you can see that we get two files. We get the data dictionary levels and the data set. I'm not going to go ahead and open the data set just because that contains protected information that you need a duck to access, but let's open the data dictionary. You'll see that we get the columns for the data dictionary only for the variables that we've selected. It has all of the columns in there. Any categorical variables that we have selected, it'll have the levels for those in the second tab. Identifier columns such as participant ID or session, and then it has the metadata for my download as well. Now, when it comes to creating and storing data sets long term, this page can get pretty messy because you might be creating a lot of data sets. It might be kind of clunky. So there are a few options. You can go ahead and archive data sets. So if you select archive, it's going to go ahead and remove that from your home page. You can always re-bring it back into view by selecting show archive. And then you can always delete data sets that are not in use anymore. Or if you are finished using a data set, for example, you want to make it immutable, you have a publication out on it, you want to store it for reproducibility purposes, you can select the three dots and select lock data set. That's going to go ahead and make it so you are no longer able to edit that data set, no longer able to delete that data set. It will essentially store it in perpetuity. Lastly, we have our pre-assembled data sets. If you come to the My Datasets tab and go to the bottom, there will be the pre-assembled datasets button. As I mentioned before, we have a limit of 10,000 variables for custom datasets. So if you ever need more than that, you can come here and select all variables, core variables, imaging, imaging, sub-studies. And then down here is a specially formatted table for all of our tabulated data. If you're ever using NBDC tools and want to access the data that way and compile your own data using R, that's going to be the pre-assembled data set that you're going to use. If you want to learn more about NBDC tools, you can go ahead and access it at the information that I'm going to show on the screen. And then the last pre-assembled data set that we have is our metadata. This is going to be a list of just all of the data dictionary that exists for the whole study, so all 83,000 columns. We can go ahead and open that. And you can see that it is quite large. And it'll have every variable. So if you ever want to explore the data dictionary, but not in DEAP and just want to explore it in your own platform or just through the CSV, you can download this here and explore it that way. That is everything that I have for you in DEAP today. I'm going to hop back over to my slides now and give another plug for our documentation. So if you want to learn more about DEAP, we have extensive information about everything from how the platform works for users that don't have a data use certification to creating data sets. We also have a lot more information on how the data sets are created and compiled. So for example, one of the things that I didn't go over is the joining of the data. The joining function operates differently when you're selecting all the variables versus just selecting variables from the additional dialog box. You can learn about that here on the screen by pausing really quick, or you can go to our documentation website URL at the bottom and read even more in depth down there too. And then lastly, to kind of round out the presentation about the ABCD 6.0 release, as well as the data use certification and other data use platforms, I have some additional resources here. So if you ever have any questions about the NBDC ecosystem as a whole, using the NBDC tools or needing to submit a ticket or ask a question, that information is up at the top. Learn more about the ABCD study, our own data release documentation in the middle, and then our data access platforms DEAP in the NBDC data access platform. The URL and the documentation for those are at the bottom. Please reach out if you have any questions, and thank you again. Here's our contact information for you.